Hopefully you can all see my pointer. So thank you very much I want to thank the organizers for a nice conference and for giving me the opportunity to invite me to present my work. It's also, I hope it would be next year. I hope it's going to be an in-person conference because we've had quite a few of these online ones. Hopefully we can go back to the face-to-face meetings soon. So I'm going to talk to you about biomolecular simulations with large-scale density functional theory, and in particular with linear scaling density functional theory with the Untitled program. So I'm going to talk about the Untitled program a little bit and solvent models, and then the type of biomolecular simulations I'm going to talk about are for free energies of binding towards applications to drug design, protein ligand free energies of binding. So I'm going to get there and show you what we have been trying to do. And then I'm going to show you some additional things that we can do with linear scaling DFT, which is energy decomposition analysis and also properties such as electric fields in proteins. And all of these concern calculations on entire biomolecular assemblies, such as proteins and other things. So Professor Clementi gave a really nice talk and introduction of the methods in her talk. So this makes my job here easier because I'm introducing you density functional theory. It's a quantum method, first principles, quantum mechanics, but based on the electron density instead of the wave function, a very well established and very commonly used approach for simulations, atomistic simulations. It includes electronic detail. This is the energy expression, and all of these terms are known: the kinetic energy of the electrons, the nuclear attraction energy, the electron-electron repulsion energy or Coulomb energy, electrostatic. But what is not known is the exchange correlation functional, and this is the difficult point of density functional theory, that approximations have to be made, and it's a point of ongoing research. And there is a hierarchy of approximations from the local density to, which includes the density of the electrons, the generalized gradient approximation that includes also the gradient, meta-GGAs, which include the second derivative of the electron density, and hybrids, which include also Hartree-Fock exchange. And as you go towards more complicated approximation, you are supposed to go up in chemical accuracy. And we will see some of these approximations later in this talk, so that's why I wanted to introduce them now. But talking about density functional theory, if you try to do calculations in density functional theory as you develop the theory in the textbook, the CONSUM approach of density functional theory, you still have to deal with some wave functions, which are one electron wave functions, and you build the density out of these wave functions. And on the process of doing the calculation of making these one electron wave functions, which are called molecular orbitals, the process has several steps where the computational effort increases with a third power in the number of atoms. So, for example, the diagonalization of the Hamiltonian is such an operation. The orthogonality that you need to impose between the orbitals is another such operation. So inherently, density functional theory has this scaling. As you increase the number of atoms, the computational effort scales rather unfavorably. And if there is a need sometime to do a calculation on a nanostructure or an entire biomolecule with conventional density functional theory, this is a serious bottleneck that you would be basically wasting computer time if you're trying to throw more and more resources if you have this type of scaling. So the motivation for developing linear scaling formulations is this: you need to study complex systems that you want to study in their electronic detail, and we need to have density functional methods where the computational effort scales linearly. So this has been an area of research that, with many developments, the linear scaling DFT methods have been developed for 30 years or so, And it's an area which proved very difficult at the beginning because we are working with conflicting requirements. Linear scaling requires localized wave functions, requires the nearsightedness principle of electronic matter, the principle developed by Walter Kohn. But on the other hand, when we do density functional calculations, we want large base set accuracy, and this typically is obtained with extended functions, extended orbitals, and so on. So it has taken a long time and a lot of developments, which I'm completely hiding here, 20 years of work, to develop linear scaling approaches where the computational effort is linear, but also we retain the same high level of accuracy as conventional DFT. And Wanted is such an approach where we achieve this by having two levels of resolution. So we have a fine level of resolution, which is in terms of a base set, which is very detailed, like a grid or a plane wave basis set, in which we express a second level of resolution, which is localized orbitals, which we call them non-orthogonal generalized Wannier functions. So these colorful shapes here are the NGWFs. So these are optimized inside the calculation, inside the chemical environment, and the coefficients that connect them are called the density kernel. This is the coarser level of resolution that connects this minimal set of generalized Wannier functions. So just to show you what this means in practice is that it means that you get with localization, you still get high accuracy in your calculation as conventional DFT. These are conventional DFT calculations with huge base sets of interaction energies, and this is what we get from our calculations. We get agreement to a conventional large base set DFT calculation. The ultimate basis set here is called periodic sinc functions, and they are essentially they're made of plane waves, so you get what is called plane wave accuracy. Now, this is the end result where we can see the scaling. With such a method, we can go to tens of thousands of atoms and calculations on lipid membranes of increasing size, fragments of lipid membranes, linear scaling DFT, and cubic scaling DFT. You see the bottleneck that is very limiting. So with an approach such as Wanted, we can do calculations on big things where we need both the accuracy of DFT and the complexity of a big model, a realistic model. So another thing that we need to include in some of these calculations is the solvent. And for this, you can either do sampling of solvent, but then this is very demanding in terms of configurations because we have solved the linear, the length scale problem, but not the time scale problem. So you can only do sampling in initial molecular dynamics of a few picoseconds, which is often not sufficient. Or you can use an implicit solvation model, and we have developed very efficient and very accurate solvent models in Wanted, which describe the solute, the solvent as a dielectric interacting electrostatically with the solute. And for this, we have a cavity, which for the solute, which can either be electron density contour based or geometric soft sphere based. And from this, we get a free energy observation from the electrostatics, but we also get contributions from non-electrostatic terms. So having all this machinery in place, we are looking at biomolecular simulations, and one challenge that is very interesting to look at is the challenge of possibly improving drug design. So in drug design, there is this computational drug design before you go to experiments in order to help experimental work. There is what they call this discovery funnel, where you have, for example, a target protein, a target biomolecule, and you want to see how certain candidate molecules bind. You want to see the affinity and selectivity, and you start with huge amount of compounds, and you screen them with methods of increasing refinement, increasing accuracy, and increasing computational cost. And you can see here the hierarchy of methods. And the challenge here is, can we use DFT as the most refined of these methods? Because it will show advantages over force fields, because it's more transferable to new compounds. It doesn't need explicit parameterization to go to a new type of molecule. So if we can show some improvement of the order of kilocalories per mole, which is what we call chemical accuracy with DFT, that can be a big achievement in the area of drug design. So we started with a simple example. This is the T4 lysozyme double mutant protein here. It has about 2,600 atoms. It has one small polar binding site. And we chose this, our collaborators in Boehringer Ingelheim chose this because it is well known, well studied. It is a very easy protein for them to start with. And we follow an approach to calculate free energies of binding in solvents. So we need to have a thermodynamic cycle, as you can see here, and the end result is that we want to get is the delta Z of binding in solvent of different ligands to the protein. And we do this with an approach called MMPBSA when you're using a classical force field, or we can call it QMPBSA when we're doing it with DFT. So it involves classical molecular dynamic simulations from which, in explicit water, from which we extract snapshots. And then we compute the free energy of binding using implicit solvent for each snapshot, and then we average all these snapshots to get the converged free energy of binding. It is not the most rigorous statistical thermodynamic approach, but it is used quite a bit in the free energy calculations community. There's more advanced approaches, of course, and we will talk about this later. So what we have done here is to try a number of ligands that you can see here. And we have tried a number of methods that you can see here described by the names PBE, VV10, and so on. And we have added on 50 snapshots with different methods for most of the ligands, or 100 snapshots with a subset of the method. So first, we looked at sampling, and then we compared the different methods once we determined that we have sampled enough snapshots. So here is the effect of the sampling. So you can see here that PBE, which is GGA exchange correlation functional in DFT, converges more or less in the same, I mean, not in the same amount, minimizing the error in the same amount, but converges in the same rate as the classical force field and this approach, which is a tight binding approach. So what we get from this is that when we calculate the enthalpy of binding from this, we can converge, we reach convergence with about 50 snapshots. So 50 snapshots are sufficient. Now, another thing that we do here is to include some entropy, and this comes from vibrational entropy from each snapshot. And this is something surprising that we obtain here, is that for a particular enthalpy calculation, here it's 100 snapshots for the enthalpy, you see that there is a big variation, a big error if you use only a few snapshots for the entropy. You have to go to about 50 or more for the results to plateau and converge. So calculations that do something and then add only 1 or 2 snapshots of vibrational entropy are not going to be very reliable. So these are the methods that we tried in this study. So we have a classical force field, PBE, which is an exchange correlation function, sorry, a GGA exchange correlation functional. PBE augmented with a few of the empirical dispersion models. VV10, which is a more advanced functional, is a fully non-local functional, which includes dispersion directly in the functional rather than empirical. And this one here, B97MRV, which is a meta-GGA, which includes also the non-local VV10 inside it. It's a highly parameterized mixed functional that has been calibrated on thousands of molecular systems, and according to its author, it's supposed to be amazing. And this is a Dftb approach, which is based on the second moment approximation. Is an available, is one of the Dftb approaches that are available. The advantage it has is that it comes ready with parameters for all the elements. And now we can see the results of evaluating all these methods. And I'm not showing you for every ligand separately because we don't have time in this talk, but this is the RMSD with respect to experiment for the free energies of binding of all the ligands that we tried with the different methods. And what we observe here is that MM has a value here, and then we compare this with all the QM methods, and they don't get too much lower in terms of error. So the best we can do is when we use PBE with dispersion. Actually, I had the PBE without dispersion, but didn't include it here. It's much worse without dispersion. So PBE with dispersion, in the best case, gives you some better results, and you can see it here also. It is consistently consistent. The PBE with dispersion is consistently better, but not dramatically better. But you could say consistently. And this is dispersion, and the ABC term is a three-body addition to dispersion. The other surprising thing is that VV10 is not as good, and neither is the highly parameterized multi-combination functional B97MRV. And also, perhaps not surprisingly, the Dftb approach is the worst of all. So some mixed results, and we need to consider why here, what's happening. I will tell you in a moment, but before we go there, I want to discuss the computational effort of this calculation. So you can see what I'm writing here, but the bottom line is that these calculations, yes, they are much more computationally costly than if you were doing it with force fields, which you can do it on one workstation. But if you add all the work that we did, and here it is not just one production simulation, but it's several because we had to compare different functionals and so on. But the entirety of this work can be done in less than 24 hours if you had in your disposal the resources of a tier 0 facility such as the Hawk supercomputer or the entire Archer 2 when it is ready. So it is not something that is unreachable anymore. It's something you can do if you have a problem that will benefit from this. It's something that is quite doable. And obviously, you're not going to get the entire Archer 2 for 24 hours, but you will get time on Archer 2 to do it in a few days or weeks. So,
sorry, you have about 5 minutes left.
Okay, thank you. So in terms of the conclusions from this study, what I want to say is that DFT shows the promise of higher accuracy and transferability, but T4 lysozyme is not was chosen as a starting point because it's a well-known protein, but it's not ideal for this purpose because all the ligands that we tried are simple molecules that are well described by force field. The advantage of DFT is when we go to ligands that are more exotic, they contain halogens or other functional groups that the force field is not well trained to represent. So this work would benefit with more complex proteins with chemically more challenging ligands. So this is what we're doing next. Also, perhaps other functionals, because the functionals we tried, the GGA, PBE, with dispersion was the best, but other functionals, maybe such as hybrid functionals, which we have and are fast now in Wanted, would work, would show further advantages, or even DFT-derived force fields by machine learning, for example, or fitting. And also more rigorous approaches that should be used or can be used based on better configuration of sampling, such as free energy perturbation or hybrid Monte Carlo. We have done such things in the past with calculating free energies in explicit water with consistently improved results compared to the classical approach. Now, in what remains, I want to show a couple more applications of large-scale DFT. One of them is energy decomposition analysis, where you can take, and why I'm doing this is because when you do large-scale DFT, you do a lot of calculation work to obtain energies, but also you obtain a lot of other information which you can use to benefit further, including energies and wave functions, electronic densities, and so on. So here we can see an approach called energy decomposition analysis, where the binding event, the association of two biomolecules, for example, can be split into stages, and we can see how much each stage contributes. So, for example, first you have the frozen density, and you can get electrostatics and exchange repulsion contributions. Then you have the polarization stage, where the density is allowed to polarize, but not transfer from one fragment to another. And then the charge transfer state, where everything is allowed to equilibrate. So the interaction energy can be broken into these components. And here is an application in the thrombin protein, which is much bigger with much more flexible ligands, as you can see here. And we've done quite a bit of work here, but I'm going to show you only a very simple, very small example. So here are two ligands that have essentially the same free energy of binding, not free energy here, but they're very different. So, for example, ligand 1 has a much larger repulsion, Pauli repulsion, steric repulsion, than ligand 2, but then this is counteracted by the other contribution. So why is this important? It is important to a medicinal chemist who wants to study a ligand with particular types of properties and interactions. Other thing we can do here is to visually understand how these interactions happen. So here we can do electron density differences. So this is the difference in polarization when you allow the density upon binding to polarize but not transfer. And then the second electron density difference for charge transfer, where you go from polarization to electron transfer. And these are the two ligands, ligand 1 and ligand 2. You can see where the interactions happen for each ligand, and then you can subtract these two images, if you like, if the ligands are similar in terms of their functionalities, and you can see where the main differences are between the ligands. That one ligand, that the ligands interact in a different way with when they do the polarization interaction, and then where is the difference between the ligands when they do the charge transfer interaction. So that's one extra thing I want to show you about DFT. The second extra thing is that we can use it as a method for validating certain force field approaches. This is here in the cyclophilin A protein enzyme, where the catalysis mechanism involves what's called an electrostatic handle that during the transition state makes a trans to cis isomerization. This is supposed to happen experimentally, they think, via alignment of the carbon monoxide dipole moment with the electric field inside the protein. And what we did here was to use Wanted as a reference, the DFT as a reference, and compare a polarizable force field with a couple of fixed charged force fields. So and calculate the electric fields within the active site. So here we have done calculations on snapshots of the protein with the surrounding solvent, explicit solvent, with about 20,000 atoms for each DFT calculation on 30 snapshots. We have the Wanted fields here, And then these are the molecular mechanics force field, the molecular mechanics electric fields. And we can see that amoeba, which is the polarizable molecular mechanics field, agrees better with DFT than the other ones. So at least this shows that some force fields are made to be to describe the physics better because they have the advantages here, such as the polarizability. It's not just energies that are the criterion for biomolecular simulations. There's other properties we need to consider. So just to finish here, I have shown you DFT calculations on entire biomolecules with wanted. These are becoming feasible and we can be used for routine work. I showed you the T4 lysozyme protein work. It is not the ideal protein to show the advantages of this force field, the advantages of large-scale DFT. So future extensions of this work will focus on more complex assemblies, but also more complex biomolecules that have interactions that are not well described by force fields, but also on more rigorous statistical mechanics approaches for the free energies. And also entire biomolecule calculations can give you, with DFT, entire biomolecule calculations can give you a host of important information, not just energies, but also other quantities can be extracted, such as energy decomposition analysis or electric fields. So just to finish here, acknowledgements to my group and collaborators and industrial collaborators. The people with red here are the people from my group and from my collaborators who have contributed to the things I showed you here. So thank you very much for your attention.
Thank you very much for the very nice talk. And we have time for a few questions only. So if you could please enter your questions in the Q&A or just raise your hand. That's even easier if you just raise your hand and then you can ask the question. And in the meantime, maybe let me ask a question. Because I think also conformational sampling is very important for this protein. So how do you set up the starting conformations that you're using in these simulations?
For the T4 lysozyme work?
Yes, for example, yes.
These are long molecular dynamic simulations of, tens of nanoseconds, and they're well equilibrated, and then we sample, in this case, we sample equally spaced intervals. So
I see. Okay.
More sophisticated approaches, such as hybrid Monte Carlo, you can select snapshots that correspond to the quantum ensemble and not directly taken from the classical ensemble. Or free energy perturbation, you can generate the energy mutation from the classical to the quantum ensemble. But yeah.
Yes, thank you very much. I don't see any questions in the Q&A. I don't know if you see if the people attending see where the Q&A questions could be entered. It's next to the tab next to the chat. And also raised hands, you could just raise your hand. But if there are no questions, I think if there are no questions that you can come up now, right now, then I would suggest that we have one raised question. Yes?
Hello? Yeah, hi. Well, thanks for the talk, Chris. I was wondering, when you show the comparison between the calculations done with one tap and many force fields, so amoeba and non-polarizable force field, what's the metric you used to make this graph?
So here we looked at, the force field is a vector. So the electric field is a vector. So we looked specifically at X, Y, and Z components of the electric field at particular atoms on top of the atom sites, inside the active site. So we compare every X, Y, and Z component of the electric field on particular atoms, the atoms that we're interested in on the active site.
Yeah, sure. So basically, it means that you look at a particular site and see what's the local electric field as compared to your reference calculations. Is that right?
Yes, I mean, on particular atoms, you can look at, yeah, on as you said, yes.
Okay, thank you.
Yeah.
Thank you very much. And I think at this point, yes, so there is just a question if you could, okay, put the slides in the slideshow. So I'm not sure about that question, but I think maybe we could proceed to Umberto Terranova. Thank you very much, Chris, again, once more. Thank you. Yes, and yeah, and so it is a pleasure to have Umberto here, who will talk to us about how carbon monoxide dehydrogenase converts CO2 to carbon monoxide. So.
Okay, thank you very much, Edina. I hope you can see my screen. My name is Umberto Terranova, and I am a lecturer at the University of Buckingham. Today I will present my latest research on how carbon monoxide dehydrogenase converts the CO2 molecule into carbon monoxide. This is the outline of my presentation. I will first introduce the enzyme, the carbon monoxide dehydrogenase enzyme, in the context of CO2 conversion. Then I will mention a little bit the methods that I used for this investigation. I can anticipate that this was a molecular dynamics study that made use of ab initio metadynamics for the calculations. And next I will present the results. In particular, I will first talk about how the CO2 molecule binds the active center of the enzyme. And then I will present the exploration of the free energy landscapes of the reaction mechanism, which I have used finally to determine the catalytic mechanism, which includes some of the experimental findings as well. In the last part of my talk, then I will conclude. So let's start with the ever-growing CO2 emissions problem. We know that there is now overwhelming evidence that the main cause of the and of that we is the conversion to as can feedback talk instead. For the motifs able to CO2 force to bring down
I'm afraid that we are not able to hear you.
The COD.
Sorry, I think, sorry, Umberto, but I think that we are not able to hear you. Maybe there is a problem with your internet, potentially. And now we have lost your, yes, we couldn't hear you, and now we don't have, so your internet seems to be very bad, and now we don't see anymore your screen. Maybe you could try to switch to the low definition mode on your AirMeet.
I've done it. Is it any better?
Yes, it seems a little better. And yes, but you have to unfortunately share your screen, so we cannot save much by switching off the video, I think. So
am I sharing now?
No, you're not sharing. I think your internet was not enough, not good enough to share. So can you try to share again?
Yes.
Okay, and I think now we were able to hear you. So let's try to continue, and in case we don't hear you well, we
Okay. Okay. I will continue from this is where I was. So the CODH is a homodimer, and the two subunits are bridged by iron sulfur cluster, which you can see here in the middle of the representation. So you should think of the two subunits as a top and a bottom. Each subunit contains an additional iron sulfur cluster and a nickel iron sulfur cluster at the farthest from the central one, and in one of them you can see in blue a nickel atom. These two iron sulfur cluster containing nickel are also known in the literature as C clusters. It is in the C cluster that the reduction of carbon dioxide occurs. The C cluster consists of a cubane cluster that is bridged to a peculiar and external iron atom. Now, so far, the reaction mechanisms at the cluster have only been speculated by using X-ray diffraction structures. That's why it would be important to have a detailed knowledge of the reaction path, of the reaction mechanism, because this could potentially pave the way to low-cost and bio-inspired synthetic catalysts that use the most important features of the enzyme. So, as I mentioned before, the study was a investigation that made use of the energy landscape of the reactor. The step was to create the enzyme binding tool and use all the cluster. Model contains 180 codes, and I will check after the structure the model, first of all,
Umberto, I'm afraid again we are not able to hear you very well.
The center shown on the left. These two molecules to the center, they are shown as well to close to the C class. Is one is this sequence, and one check interact by bonds with the CO2 molecule. One important fact that a transfer occurred from the histidine to the CO2 molecule form a COOH ligand that has never been reported by studies, and these are the highlighting of the chemistry of molecular dynamics in this study. Of this establish the stability of this structure with respect to the one where the instead as you can see here, so the exact nature of the
I'm afraid, Umberto, I'm afraid that your internet is not able to support your sharing of the screen. So we couldn't really hear you well.
This is strange because I've been teaching with sharing screen for
yes, but your internet today is somehow not working very well. So, I mean, you could try to switch off your camera, although I'm not sure it's going to help, but we could try that way. You switch off your camera and try to continue your talk.
Okay, I can try. Any better? Can you hear me?
Sorry, we can hear you, but you're not sharing your screen. So if you could please share it again.
I have to reload it.
Yes, and are you in the low bandwidth mode?
Yes, yes.
On the air, okay.
Yeah.
Okay, yes.
Okay, let's continue.
Yes.
So I was saying that the metadynamics was able to confirm that the COH ligand is more stable than one where a CO2 is present, and these will have implications for the reaction mechanism. So in search of intermediates, we spanned the reaction coordinate of the CO2 distance, the carbon dioxide reaction coordinate, the distance between carbon and O2. And we noted that when you bias the dynamics by adding hills, the CO2 bond is weakened, and a transfer of a proton from the lysine to the O2 atom occurs, and the event is followed by an internal transfer of proton mediated by N1. So there is the formation of one water molecule, and the starts. And the structure is one where the CO binds in a monodentate fashion the nickel atom. So given that we are the COH ligand, I considered also an alternative mechanism that involves the elimination of a water molecule, which is what the ligand suggests would occur first. So this time we have chosen as collective variables the O1 H2 distance and the CO1 distance. So a direct transfer of a proton from the lysine to O1. And in this case, the energetic cost to eliminate the water molecule was only 0.6 eV, after which the system was able to escape the minimum you see from the figure on the bottom. The resulting structure is one where nickel is in a distorted square planar geometry, and the carbon monoxide is now in a bridging configuration to the cluster. Nickel geometries, planar and the that I mentioned before, have been observed experimentally. So we suggest that the key factor determining the geometry of nickel is the reaction pathway. In other words, which oxygen atom of CO2 is eliminated as part of the water molecule will determine the subsequent geometry of nickel. The free energy required for the carbon monoxide to diffuse was calculated again by another dynamic simulation, and this was about 0.9 eV, which is 0.2 eV lower than the previous path. And you can appreciate this in the figure above. So with all the information from the previous energy landscapes, I was able to revise the catalytic mechanism of the enzyme. And what you can see here is what I suggested. The cycle starts with CRED2, which corresponds to the reduced structure observed experimentally by X-ray. The exchange of water for carbon dioxide leads to the formation of the COH ligand, and we have assessed already the crucial role of the histidine that is necessary to create the COH ligand. Then the lysine 563 transfers the second proton with the subsequent elimination of the water molecule in the intermediate 4. The next step is the elimination of carbon monoxide with a step of 0.9 eV to form what has been observed experimentally as the CRED1 species at intermediate 6.
Sorry, but I think the 15 minutes, okay, that's your conclusion side.
We have presented the metadynamics investigation of the mechanism of reduction of CO2 to carbon monoxide. We have found that the ligand that binds the C cluster is actually carbon dioxide, but COH. And we have shown that nickel geometries
maybe Umberto, if you don't mind, I think it might be just better if you conclude.
Terminal L.
Because I don't think we can hear you anymore again. Sorry, if you might just, yes, thank you. If you might just maybe turn off your screen sharing, and that way we would be able to hear you, I think.
Okay, so
Yes.
I was just saying thank you for your attention, and I would like to thank the Materials Chemistry Consortium and Archer 2, without which this research wouldn't have been possible.
Thank you very much for the talk, and I think we could still hear him for the most part of his talk. So if we have questions or raised hands. In the meantime, maybe could I ask you, Umberto, what was the collective variable that you used for this enzyme reaction? Because I think I missed it.
So that there were quite a few collective variables. For example, when in the simplest case, when I studied the detachment of carbon monoxide from the C cluster, the two most obvious collective variables were the two bond distances of the carbon to the nickel and iron atoms, respectively. But I didn't have time to go into the details of the collective variables, but
okay.
Unfortunately, but yes, there were a few of them.
So you were able to use multiple with the metadynamics in conjunction?
Say that again, sorry?
You were able to use multiple coordinates, multiple collective variables with your
up to two collective variables, yes.
We have another question from David Santos Carbalal, and the question is, did you consider the solvent in your simulations?
Okay, David, I didn't include the solvent because apart from the crystallographic water present in the X-ray structure close to the C cluster, because essentially the C cluster in nature is embedded within the protein, so I don't expect here the solvent to play a major effect.
Thank you very much. And we have a few more questions, so I think I'm just going to show you the questions quickly. From Davis Di Tommaso. In your reaction mechanism, are you proposing 2 proton transfer steps followed by 2 electron transfer steps? And the accepted mechanism is the concerted proton-electron transfer. Could you comment on this?
So in your mechanism, you're proposing 2 proton transfer steps followed by 2 electrons. In other situations, they said negative conserved proton-electron transfer. Thanks, David. Davis, I don't know which other mechanisms you are referring to. In this case, I only investigated the proton transfer followed by the electron transfer because of the presence of histidine that would naturally act as a base, and the same for the lysine. So I haven't actually investigated a possible concerted proton-electron transfer.
Yes, Davis, is that answering your question? You are muted.
Sorry, yes, it's answering my question. Thanks a lot.
Thank you very much. And then we have Marco. Just a second. I think it's already answered. So basically, we have, I think, no more questions, if I see it correctly. Thank you very much again, and see you in the next session in about 5 minutes.
Thank you very much. Bye.
Bye.